was GOS2. Now GOS2 is the G0, G1 switch in the cell cycle. As it turns out, it's also a negative regulatory factor of ATGL. And what this ATGL complex does is it takes in lipid molecules from the cell and it turns them from triglycerides into diglycerides so they can go in to acetyl-CoA and go into the mitochondrial respiration and create active ATP and reactive oxygen species. So we wanted to find out what happens to GOS2 as we age. So we use fibroblasts from two cell lines. One cell line has fibroblasts at 3 different ages, 3 different individuals. So 3 days, 12 years, and 61 years. So we grew these cells out till they reached quiescence. So when they're in quiescence, they're in that G0 stage. And we collected them for QRT-PCR to measure their mRNA expression. And what we found was as we age, our GOS2 actually decreases quite significantly. This is important because if GOS2 is decreased, then this is allowing this process to function and go through and feed into mitochondrial respiration. This is actually really interesting because we saw this shift from mitochondrial respiration, from glycolysis to mitochondrial respiration. So our results show that it's actually as we age, we're feeding more into this mitochondrial respiration. So our results fit perfectly. So then we also wanted to study it from the same individual because we want to have more consistency. And we found the same results. So this is fibroblasts from 29 years old, 36 and 46 from the same individual. And we saw the same decrease in mRNA expression. So then we wanted to look at, just as a bonus, okay, if this is the negative regulator, let's look at the positive, the co-activator of ATGL. And we found as we age, it increased. So that was good. So we also looked at ATGL. Didn't really change much, and this makes sense because this is a protein-protein interaction. GOS2 doesn't negatively regulate it through a transcription factor through the DNA. It's protein interaction, so we wouldn't really expect to see a change in the mRNA. But as a future experiment, we want to look at the protein activity. And we think when we look at the protein activity, we'll actually see an increase in the protein activity of ATGL, a decrease in the protein activity of GOS2, and an increase here. So these are actually really good results. GOS2 it's everywhere in the cell. It has a lot of influence and a lot of processes, so it's part of the cell cycle, regulating whether the cell's going to grow or not, but it's also part of this lipid metabolism lipolysis. So really significant results here. Can play a role as therapeutic targets for a lot of different diseases such as cancer and also non-alcoholic fatty liver disease. Really cool.
I have a question. What do I miss here? No?
I have a joke.
I'm sorry.
So yeah, in the future, we're going to look at the protein activity by doing Western analysis. We're also going to manipulate the genetics of GOS2 if we want to knock it out and see what happens to the cell, see what happens to the expression. We also want to do an overexpression of GOS2, see what happens. So we're really focusing on lipid metabolism and how that is affected during aging because of this age-associated metabolic reprogramming that occurs. And hopefully this will tell us more about the cell as it's aging and why this influences the increase.
When did you start the research?
So actually this research started about a year ago, and that's where these results came from. They did a metabolic assay of obviously a lot of metabolic genes, and from there we kind of targeted down to these major ones that we saw a major change. And then I came in and found the change in GOS2. So these are all basically in the mitochondria genes, so they have to do with metabolism, mitochondrial respiration.
This part is what you did this summer.
This is what I did this summer,
Gotcha.
and this has to do with lipid metabolism.
Gotcha.
So this is with mitochondrial respiration and glycolysis. And we also found that lipolysis, lipid metabolism, is affected as we age.
Very good.
Yeah. And we'll also, another future experiment is when we do these genetic manipulations with GOS2, we're actually going to take these genetic manipulations in young and old fibroblasts and put them into tumor cells of mice and see how it affects the tumor. So we're going to inject these old and young fibroblasts into the tumors of mice and see if it increases their proliferation or if it decreases their proliferation. So hopefully next summer, next summer, it'll be really interesting to see those results.
hip hop, yeah.